scientists have found the key to artificial life after creating yeast from man-made DNA. The synthetic yeast could be used to create painkillers, biofuel, bread better able to withstand high temperatures and cheaper beer for the brewing industry. More importantly, yeast shares a quarter of its genes with humans, so may allow biologists to build whole sections of DNA to prevent devastating diseases like cystic fibrosis. An international team of scientists successfully replaced almost a third of the genetic material in baker's yeast after creating 5 new synthetic chromosomes with DNA made from chemicals in the laboratory. The team, including researchers from Imperial College in London, yesterday announced they plan to create entirely man-made yeast by the end of the year. The breakthrough will reignite the debate over GM food, as artificial yeast could in future be used to make bread more nutritious. It also raises fears over bioterrorism, as the same DNA swapping methods could be harnessed to create extra deadly viruses. However, the researchers involved say it is a major milestone in the race to create the first fully artificial complex living organism. They not only inserted new DNA into yeast but removed that which was unnecessary, showing scientists can speed up evolution by taking the building blocks of life and rearranging them. Professor Patrick Kai of the University of Edinburgh's Wellcome Trust Centre for Cell Biology said, "We are excited to mark this milestone in our quest to create an entire organism from scratch. The yeast genome is like a book, which in yeast has 16 unique chromosomes, or chapters. It is as if we took a paragraph out, rewrote it and put it back." Professor Paul Freeman, who was not involved in the research but is co-director of the Center for Synthetic Biology and Innovation, said, "This work represents an amazing advance in our ability to chemically synthesize the blueprint of life." He added, "The fact that yeast cells with different synthetic chromosomes grow normally also shows that evolution has provided us with only one blueprint for life, and through this work and other work around recoding genomes, we are now beginning to realize that there may be many different genomic blueprints for life." Scientists first created life in the laboratory 7 years ago, when a single cell of bacteria was produced by a U.S. team at the J. Venter Institute. 3 years ago, the first synthetic yeast chromosome was made, comprising 272,871 base pairs, the chemical elements which make up the code of DNA. Now biologists from universities around the world, including Edinburgh University, Imperial College in London, Johns Hopkins University in Baltimore, the Sorbonne in France and BGI in China, have separately built synthetic strands of genetic code letter by letter. Each combined, stripped of non-essential DNA, caused yeast to grow normally when swapped with its genetic material. The artificial version could be used to make yeast for bread baking, which is more resistant to high temperatures, or make brewing cheaper through yeast, which is more tolerant to alcohol. It could also be used to manufacture painkillers, antibiotics and biofuels, which separate studies show could replace petrol and jet fuel. Baker's yeast is similar to human cells but simpler and easier to study. While the largest synthetic chromosome created so far is still 1/3000th the size of a human genome molecule, it could lead to gene therapy for human patients. Synthetic DNA, created in the same way, could be used to replace faulty DNA, which causes diseases like cystic fibrosis and Down syndrome. Professor Jeff Boak, part of the Synthetic Yeast Genome Project, said, "We imagine this kind of technology could be used to usher in, for example, a new era of gene therapy where we could deliver not just a single gene as is done currently, but could think about bringing in whole networks."